Hello lovelies, in this video we're going to be going through your AQA Biology Paper 2 2023 predictions for your Combined Science Higher Trilogy. If you're doing one of the other specifications or you're doing separate science, then look in the description down below for links to those paper walkthroughs. Here we're going to be taking you through what the command words mean, exactly what sort of things should go in an answer. We're going to be teaching you all the science, if there's anything in there that you're not 100% sure about, and then we're going to be talking about how you can structure your answer to get the most marks.
Hi everyone, and welcome to this video where I'm going to go through the AQA GCSE Combined Science Biology Higher Paper 2. So hopefully you've done the Higher Paper 1 for the Combined Science, and this is you having done the Paper 2, and now you're going to watch this video with me. I'm going to go through each question step by step. I will help analyse the questions, find clues in the questions, help you give you some exam tips of how to tackle these questions in the real exam, give you the model answers, and then explain the mark schemes, because I know they can sometimes be tricky. So hopefully you've had a go at this paper and you've either marked it and you just want some help with the marking, or you're going to mark it along with the video. That's fine. And just remember, we've got the grade boundaries down there. They're very rough guides, so they're not going to be guaranteeing you the same grade in the exam, but it just gives you an indication of where you are at with your percentage-wise. Okay, so have your paper ready and off we go.
Okay, so first question. The image shows 3 dogs. They are all members of the same species. What is the definition of a species? So this is a 2-mark question. It's just a definition question. Hopefully you know the definition of a species, but just to be clear that we've got to have enough detail in there to get the 2 marks. So we need to say that they're similar organisms and that they can breed, but we need to make sure we also say they can breed to produce fertile offspring. Because remember, things like lions and tigers are similar. They can breed, but their offspring are not fertile, which is why lions and tigers are therefore different species. So a group of similar organisms that can breed to produce fertile offspring. The image shows different breeds of dogs that look very different. What term is used to define the physical differences between dogs? So basically, what is the word that we use to describe the physical differences between organisms? So when you look at all of the people in your class, everyone looks slightly different from each other, even though obviously we're all from the same species. What are those differences called? Or what's the cause of those differences? It's variation. The table below shows the classification for dogs. Complete the table. Okay, so we've got a classification table or a taxonomy table, and we need to fill in those gaps for the names of the groupings. So hopefully you can try and remember the sequence. I like to use king, prawn, curry, or fat, greasy sausages as a way of remembering the letters in order. And then we just need to remember what those letters stand for. So the K is for kingdom, the O is for order, and the S is for species. And then obviously we have phylum, class, family, and genus already done for us. So we just need to make sure we've put the right words in the right order. So yeah, use that and like an analogy to kind of remember K, P, C, O, F, G, S. Just remember the right order, and then it's just about remembering what those letters stand for. Give the binomial name for dogs. Use the table above. So hopefully we remember the binomial name means 2-name or 2-part name, and it comes from using the genus and the species names from the table or from their classification. And so the binomial name for dogs, its Latin name, is going to be Canis familiaris. And we need to copy it exactly as it is in the table. So capital C for Canis and a lowercase f for familiaris, because you always leave the species name as a lowercase. Okay, part E, give one reason why the binomial name is used instead of conventional names and why they're used by scientists. Well, the binomial name is that Latin name that we just looked at from the classification, and then a conventional name would be kind of dog, for example. The first thing that I kind of think about with this question is, well, if we didn't use this Latin name, if all of the scientists around the world didn't use this Latin name and we were using the conventional names, they would be different in every language. So think like dog, chien in French, it's different in every language. So you would have to understand what was meant by that word in the language with the person you were speaking to. But if we all use this one Latin name, there's no confusion. It doesn't matter where you are around the world. And also the fact that we've got two parts to it, we've got the genus and the species. The genus can actually tell you the evolutionary relationships of that animal. So if I know something else, like the wolf, for example, has the genus Canis, then I know that they're very closely related to each other. And that can be helpful when you're kind of looking at different species or species you're not sure about or something you don't know about. Whereas if someone just says dog, that can mean specific things to specific people. So I would go with it, the same name gets used by everyone all over the world, regardless of language, and it allows us to all be talking about the same organism. Okay, part F. When the new 3-domain system was introduced, dogs were classified into the eukaryota. Explain how the 3-domain system was developed. So it says explain how. So we need reasons that how it was developed. So how was it changed? Why did it come about? So new technology, basically things we didn't have before when the first classification system was thought up. We have DNA, we have electron microscopes, we have biochemistry evaluations, which we didn't have back in the days before DNA analysis existed. And how was it developed? So what did that extra technology allow us to do? Well, it allowed us to split the prokaryotes into bacteria and archaea, because we found out that they were actually significantly different from each other. It can't just all be lumped in together as prokaryotes. They had to be in their own separate domains. And obviously the bacteria contains the normal true bacteria, and then we have the archaea, which are the extremophiles. So I'm going to use all this information to get my 3 marks for this question.
Okay, so question 1, mark scheme, let's have a look at it. So you can say, instead of saying similar organisms, you can say they have the same number of chromosomes. You can't say organisms that have the same number of genes. That's not really saying the same thing, because some organisms can be in the same species but not exactly have the same number of genes. Do not accept mating instead of breeding. You really need to say breed. You can't just say mate, or you can't just say breed and then not say the fertile offspring part. For the table, you need to make sure those words are in the correct order, so it has to go kingdom, order, species in that order, otherwise it doesn't make sense. For 1D, you have to have capitalised canis and lowercase for familiaris, because that is how we write those, and you are just copying from the table, so it should be in the correct case that is used in the table. For 1E, like I said, you can say regardless of your language or all over the world, they use the same name. And if you didn't say evolutionary relationships or ancestry, but you've just said to work out whether a species is closely related or not, that's also fine for 1E. And then for 1F, you needed to say based on DNA or biochemical evidence, you need to say what happened to the prokaryotes were split into 2 groups or 2 new domains. And then in theory, you should talk about the 3 domains. If you just mention them, that's fine. If you didn't get any other marks, you can get 1 mark for just saying archaea, bacteria, and eukaryota. But realistically, you should talk about the 3 domains and what they include. So the archaea being primitive, simple bacteria. You can also say extremophiles there for number 3. Or if you said the true bacteria are just modern or normal bacteria, and then eukaryotes, if you mentioned them, you would need to say includes plants and animals and fungi and protists, or just say includes everything else that has a nucleus in the cells. That's fine. But you need to kind of think about what you can actually pick, where you've picked up marks. Because you can have only 3 of those, but you don't get a mark for just saying the name of the domains.
Okay, the image shows a cranberry plant. The cranberry plants grow in bogs, which are often flooded. So this might be a clue to something we need later on. It's information about a plant that we've never necessarily seen or been talked about before. This idea of being in bogs, flooding, thinking about peat, for example, but this idea of waterlogging. So lots of water hanging around marshy, kind of wet soils. And that can obviously be a problem for some plants. If it's too wet, they won't get oxygen to their roots, and then they can literally drown. But these plants are obviously clearly adapted to live there. Cranberries have some specific adaptations for each of the following. Explain how this helps the plant to survive and reproduce. So these reasons, because we have to give reasons and it's asking us to explain, we need to give reasons and link those reasons to this idea of either survival or reproduction in order to get the mark. So bright red fruit, a fruit of any kind, but colourful fruit, is clearly there, and hopefully we know this from all the way back in Key Stage 2, I think, that it attracts animals. who will help to disperse the seeds. So the point of having brightly coloured fruit is that you want to attract organisms that are going to come and eat your fruit and then help with dispersing the seeds, either by dropping them if you're a bird, or sometimes they pass through and they get pooped out by animals far away from the plant in order to then grow somewhere else. So that's helping with reproduction and survival without competition from your offspring. Can live for up to 6 months without oxygen to their roots. So that's talking about a link to what we said at the beginning with this idea of living in this boggy, often flooded area. So if they can live for 6 months without oxygen to their roots, it means they're able to survive the flooding. They can survive and live in the bog, in the habitat, even without oxygen. One stem of a cranberry plant can produce a new plant. Okay, so this is asexual reproduction. It means that the plants can reproduce themselves fast, quickly. They can reproduce themselves or clone themselves if sexual reproduction isn't possible, which means that they can still survive and carry on. Even if they can't do sexual reproduction, they can just clone themselves and survive. So hopefully that was fairly an easy question, as long as you can kind of make sure your answer is related to survival. Features of plants have been changed over many years as farmers have bred them for specific characteristics. An example is broccoli, which is the result of hundreds of years of breeding wild cabbage plants. So this is clearly a selective breeding question. We can see it's written there. So we've got the wild cabbage that has these kind of yellow florets, big leaves, really tall, skinny stems. And then we have what broccoli looks like today. And they're the same species, they're just a different variety. So over the time, we've changed them through selective breeding, but it's still the same species, it still has the same genes. So the parts of the broccoli that we eat are large flower buds with thick stems. The original wild cabbage plants had small flower buds, thin stems, and large leaves. Describe the process of selective breeding that farmers would have used to produce modern broccoli plants from the original wild cabbage plants. Okay, well, that is interesting. So that first part of that paragraph there is telling us the characteristics that have been selected for, the desired characteristics that we have clearly selected for, that have changed the broccoli over time to have these thick stems and large flower buds, which is now what we eat. So because they've given us that information, we're going to need to make sure we use it in the question. So when we say select the desired characteristics, it's told us what they are. So we should try and use them in our answer. So let's just briefly recap the steps of selective breeding that we would need to write down for any selective breeding question, not just this one. And then we can have a think about how we're going to turn that into sentences. So this is what I do for any long answer question. It's a 4-mark question. Anything where you've got a process and it's outlining steps in order, make sure you write these down, like do a little brain dump before you write out the actual answer. So select the characteristics that you want, breed the animals together, select the offspring of that cross that have the characteristics that you want, breed those together, repeat over generations, okay? Because it takes a long time to do this. It doesn't just happen the first time. We've said this took hundreds of years. So we need to make sure we say about repeating as well there. So that's the kind of backbone of our answer. Now we just have to turn it into sentences that make sense for this question. Okay, so I've put choose wild cabbage plants with the thickest stems and the largest buds. Breed them together. Choose from the offspring that also have thick stems and large buds, and repeat this process until all of the offspring have the characteristics that we want, i.e. thick stems and large buds. Okay, so part C. Name 2 other desired characteristics in plants that have been selectively bred, for. Okay, so this is kind of moving away from broccoli now, moving away from kind of what they were talking about there. We're thinking about specific things or features that we should know or we have learned that have been selectively bred for in plants. So one of them is resistance to either disease or pests. And one of them is like large or unusual or very heavily scented flowers. One being useful and helpful for like crop plants, and one for more aesthetic reasons for flowers that we'd like to look at. So some major crop plants which have been highly selected for are at risk of being lost to diseases, explain why. So this is saying, give me a reason why selective breeding can mean that some crop plants can be susceptible to new diseases. So obviously if we've selectively bred them to be resistant to one specific disease, they will be resistant to that. But this is more about kind of new diseases that crop up, new pathogens. So why might selective breeding cause this to happen? Well, if you selectively breed over and over and over again, many generations, then you're going to have lack of genetic variation because you're trying to keep the same characteristics in. You're not mixing as much. So your genetic variation or genetic diversity is going to be low in the population, which means there's less chance that they'll have resistance genes that mean that they can be able to survive a new pathogen or disease. So these crop plants have low genetic variation, so they are less likely to be resistant to some new diseases or some diseases. That's all we need to say.
Okay, so asking for question 2. For the first 3, you have to have a valid explanation that links to survival. So you can't just say attracts animals and don't explain. You need to kind of talk about survival or reproduction in your answer. For asexual reproduction or you can use cloning. You not needing to find a mate or not needing to undergo sexual reproduction. All of those things is fine for 2A part 3. And the 4 marks for the selective breeding, you can say cross-pollinate instead of breed, or to kind of make them together. You can say that you would breed repeatedly until they bred true or until they produced modern-day broccoli for the last point. That's also fine. We talked about, so for large unusual flowers and disease resistance, there are 2 things. You can also say pest resistance. You can say heavily scented flowers instead of large or unusual flowers, because we like to grow flowers that smell nice. And then you can say reduced genetic variation, or you can say low or less. You also have to include genetic when you talk about variation there, because it doesn't necessarily, although they will all look similar, they won't look any different. So it's just about their genes being less varied, not anything to do with their characteristics or anything. So you must have that to get that first mark.
Okay, so question 3. Around the world, millions of hectares of forest is being lost every year. The graph below shows the breakdown of the causes of deforestation in tropical and temperate regions. And then we've got the tropical areas are South America, Southeast Asia and Africa. And the temperate ones are North America, Europe, Russia, China and Oceania. So then we've got temperate and tropical bars on our bar graph. The temperate ones are the dark grey, tropical are the light grey. And then we've got the millions of hectares of lost trees on the y-axis. Hectares, if you're not sure, it's just an area measurement. So it's too big to measure in meters squared, for example. A hectare is a large area. It's often used for talking about areas of land or fields, etc. So it wants us to calculate the percentage increase of trees lost to agriculture in tropical regions compared to temperate ones. So we're just looking at that first set of bars. So this is just a percentage change question. You can get these in various different forms, normally involving some kind of graph or just some data in a table. So they want you just to work out the change, the difference between the tropical and the temperate, and work it out as a percentage of the original. So how much of an increase is there in deforestation or annual tree loss because of agriculture in tropical regions compared to temperate regions. So to work out percentage change, we do the difference. So the difference between the 2 values we're looking at, the change, and then we divide it by the original number we're looking at to compare. And then we have to times it by 100 to get a percentage. So we need to use the graph to work out our numbers to find out the change. So we've got 0.9 because it's just under and halfway between the small squares for temperate, and then for tropical it's 4.9. So we are comparing the increase from temperate to tropical. So we need to work out the difference. So we need to do 4.9, take away 0.9, divided by 0.9, which is our start point, because we're comparing the temperate as our kind of start point. So 4.9 take away 0.9 gives us 4, and then 4 divided by 0.9 times 100 gives us 444%, and it should be a whole number. So 444%. is how much it is increased by in terms of the amount of trees lost or in millions of hectares from tropical environments compared to temperate ones. So part B wants us to explain why more deforestation for agriculture and fuel is occurring in tropical regions than temperate regions. So basically, why is more deforestation happening in the tropical regions than temperate regions, but specifically looking at why is it happening for agriculture and fuel, because it's not happening due to wildfires or logging. So why would countries need more farms and more fuel? So what could be the difference that's causing this? Well, hopefully we've sort of thinking about our biodiversity and impact of biodiversity and impact of climate change. So more people, so the population increases, means we need more food. So they will need more area of land to grow food or farm food for the people because there's an increase in population. You could also say there's better standard of living, so more food is required and more heating is required for homes, so that's why. So if the standard of living is increasing or the population is increasing in tropical countries more than temperate countries, then that's why they need to be clearing more land. In the last 20 years, the annual area of trees lost to wildfires has increased from 2.52 million to 9.26 million hectares. Suggest reasons for this. So basically, why are we having more wildfires, or why are more trees being burned by wildfires? And it's in the last 20 years. So what could have happened in the last 20 years that could have led to more wildfires? And because it's a suggest question, it's probably going to be something that is your knowledge or common sense that you've got. So why would there be more wildfires? What's changed in the last 20 years? Hopefully, we can kind of link those two and think about global warming. So global warming or climate change has caused higher temperatures and more droughts, or things like just less water, less rainfall, land to become more dry, and therefore more likely to catch fire. That's what this question is really asking. Deforestation reduces biodiversity and causes some species to become extinct. What is meant by the term biodiversity? This is just a definition question again, so we just need to be able to define what that word means. So biodiversity is kind of talking about how we measure how diverse, so that means how different, how big of a range of species there are in a specific area or habitat. So it's the number or the range or the variety of different species in an area or habitat. Give one reason why it's important for humans to prevent a species from going extinct. So why is it important that we stop species from dying out and there being no species left? Well, there's lots of reasons, but one of the things I would think about is if you take out a certain species, they are in a food chain. So we know that that's going to have impacts above and below them in the food chain. And so that's what I would say is one of my reasons. Again, there's lots of reasons you can talk about, and we'll look at those in the mark scheme. Okay, so the mark scheme for question 3. We've got 3 marks for the answer for 3A. 2 marks if you left it unrounded to a whole percentage. And then if no other marks, if you just substituted the numbers into the right equation, then you can get 1 mark for that. For 3B, we talked about the standard of living and increasing the human population, but we could also be talking about less access to fuel. So if there's not as readily available fuel sources as there is in temperate zones, then that could be a reason why they need more fuel. Also, this idea of urbanisation, villages expanding, something that you may have learned in geography, can count for like increasing human population. They're increasing the space. So they're chopping down trees to make way for more towns, cities expanding, villages expanding. And then any one from a load of options for 3D, part 2 and part 3. So there's lots of options there. The only thing we can't talk about for reasons why we need to maintain biodiversity is because you can't just mention food for humans. Or this idea of if we don't protect it, then we'll lose food sources, because it's not about us and food sources, really. What we're actually saying is we're affecting other organisms' food chains, other useful substances that we might find, so drugs, medicines, resources for kind of materials and stuff. But food is not acceptable, really, because we grow food as a specific sort of set of an ecosystem that we create. It's not wild, it's not untouched. So that's kind of separate from what we're talking about here in terms of wild ecosystems.
Streptococcus bacteria are among the family of bacteria which causes illness, such as a sore throat. Streptococcus bacteria and other very simple organisms reproduce asexually. Name the process by which bacteria replicate. So it's telling you that bacteria reproduce asexually, and we want the name for the process that they divide by. It's not mitosis, because it's bacteria we're talking about, not animal cells. So we can't say mitosis, even though it's talking about asexual reproduction and cell division. Hopefully we should know this. So the way bacteria replicate is by binary fission. Literally that means splitting into two. So that's how I like to remember it. So think about fission like in physics, the splitting, and it splits into two, binary. Okay, on to part C. Streptococcus bacteria are very small, with a radius of 0.6 micrometers. Use the following equation to determine the volume of a single cell. Give your answer to two significant figures. Okay, so we've got a formula to work out the volume of a sphere. We've been given the radius, which is great. So we just need to put that number into the equation, work out the answer, and then make sure we get it down to two significant figures. Okay, so we just need to put our number into the equation. So 4/3 times pi times 0.6, which is the radius cubed. Use your calculator, put it in the calculator, make sure you've put it in properly, use brackets around the 0.6 cubed. Because you get a calculator in the exam, don't try and do stuff like this in your head, because you'll just mess up and lose marks unnecessarily. Okay, so we get 0.9047. We need two significant figures. Obviously, we cannot round up that 4, so it becomes 0.90 micrometers cubed. And you need to make sure you put that extra 0, because otherwise you haven't got two significant figures, you've only got one, and you wouldn't get the last kind of third mark.
Okay, so part D. A serious streptococcus infection requires antibiotics to treat it, but some strains have now become resistant to certain antibiotics. That's helpful, but it's not necessarily helpful to the question. What structures in bacterial cells are likely to carry the genetic information that gives these bacteria resistance? So specifically, we're looking for structures, and we're talking about DNA. So genetic information means DNA. So what structures in bacterial cells contain DNA? Well, they don't have a nucleus. And if you're thinking about structures that are specific to bacteria, then the only other structures that carry DNA inside their cells are plasmids, which are the little loops of DNA.
Okay, part E. Explain how a population of antibiotic-resistant bacteria might evolve from non-resistant bacteria. Okay, so this is an evolution question. We're explaining how something happens, which means we need the steps in a process. And we hopefully know that when we talk about evolution, we're basically just talking about change over time through natural selection. So natural selection is the process which we're going to describe as part of our answer, and that's kind of how we're going to say that they could evolve over time. So we need to think about generically for any evolution question, there's going to be a series of steps and points that you have to include, and then you just try to adapt your answer to make it specific to the question we're answering. So variation exists in the population, so there's differences between individuals in the population, and that's caused by mutations. So random mutations create this variation. Some with various mutations that will be helpful will survive and reproduce. That's really important. That means they pass on their genes to their offspring, and therefore the number of individuals with that adaptation or characteristic or whatever it is increases in the population, so increases in number until there's lots of them. So now we need to just take these bullet points and we're just going to write them out, but make them specific to the question. So we need to talk about bacteria, we're talking about resistance. So in this case, the mutation, the variation that's caused by mutation is that some of these bacteria are going to be resistant and some won't be. So the mutation is going to give some bacteria the ability to be resistant to antibiotics. They are going to survive and reproduce. They are going to pass on those resistance genes to their offspring. So more and more and more bacteria that have the resistance genes are created, compared to the ones that don't, because they will die out when they get treated with the antibiotic. So there is variation in the population of non-resistant bacteria caused by mutations. One mutation makes some bacteria resistant to the antibiotic. These individuals survive and reproduce or replicate, passing on their resistance gene. So the number of resistant bacteria increases until it's the whole population. So until you get the only ones that are left are going to be resistant.
Okay, doctors want to stop more of these resistant strains from developing. Describe and explain 2 ways doctors can reduce the chances of more antibiotic-resistant bacteria strains of the Streptococcus occurring. So we need to explain and describe. So that means we can't just give 2 ways or 2 methods they could use to prevent this. We need to explain why those methods are important at reducing the antibiotic-resistant bacteria from developing. So ways in which they can stop antibiotic resistance is to not overprescribe, so not prescribe antibiotics when they're not needed. Make sure that the patient takes the full course of antibiotics. Don't prescribe antibiotics for viruses. So we just need to take each of those and then give the reason why that can affect how this can cause antibiotic resistance if they don't do those things. Okay, so my first point is going to be make sure patients take the full course of antibiotics, as any bacteria that could survive can be resistant and they can reproduce. So I've got my point and then my explanation. And so for my second point, I'm going to say, do not prescribe antibiotics to treat viruses because antibiotics can't kill them.
Okay, so the mark scheme for question 4. So we've got binary fission, and then we've got the calculation question for 4b. Just make sure you have a whole mark there for 2 significant figures. So make sure your answer is 0.90, not just 0.9. Otherwise, it's not 2 significant figures. The 4D, you've got all of these underlined words, 1 for each mark, 1 to 4. Got to have those words in your answer in order to get the mark. So you have to say variation in your answer to get mark 1. Have to say mutation to get mark 2. Have to say that the bacteria are resistant or the mutation causes resistance to be for mark 3, and then you have to say survive and reproduce for mark 4. And then for the last question, we sort of said this, you need to have them in pairs. So the description and the explanation for 1 mark. So do not prescribe antibiotics for mild infections is what they shouldn't do, the description of what they should or shouldn't do. And then you need the reason why that's going to help prevent resistant bacteria.
The human nervous system is divided into 2 main parts, autonomic and somatic nervous systems. One of the functions of the autonomic nervous system is to control reflex responses to danger. Describe the chain of nervous responses, including the name of the neurons which result in a hand being pulled away from a sharp pain. So this is a reflex arc question. So we need to talk about how the reflex arc happens, but it says describe. So we're kind of describing the steps in the process of how we get from touching something that causes pain to pulling our hand away. This is one of those ones, it's 4 marks, there's quite a lot of marks here. I'd briefly just write down the reflex arc in the right sequence. That's going to be important here for getting marks because it has to happen in the right order. So stimulus to the receptor, to the coordinator, to the effector, to the response. But we need to make sure we talk about the neurons. It wants the neurons, the names of the neurons in the right order. So obviously between the receptor and the coordinator, we have the sensory neuron. Then in the coordinator, we have the relay neuron. And then between the coordinator and the effector, we have the motor neuron. So we need to name those specifically in our answer and to make sure they're in the right order. Remember, the coordinator here is going to be the central nervous system. The receptor is going to be a pain receptor, although that doesn't necessarily need to be specifically stated. And then the effector is going to be the muscle, the arm muscle that's going to calm away. And that's our response. So we've been told what the stimulus is and what the response is, so we kind of really only need to talk about those 3 middle ones, but we're going to incorporate it into our answer anyway. So the stimulus is detected by receptors in the skin. Electrical impulses are sent down the sensory neuron to the relay neuron in the central nervous system. The impulse then travels through the motor neuron to the muscle, which is the effector, and contracts to move the arm. So there I've said receptor, I've said the central nervous system, I've said the effector, I've also talked about the sensory neuron, the relay neuron, and the motor neuron, and all of that was in the right order. And I've explained how the muscle, how the effector causes the arm to move away. So all of that should get me my 4 marks. Suggest how a reflex response, such as pulling hand back from a source of pain, might be an evolutionary advantage to a species. So why are reflexes or reflex actions important for survival? So because an evolutionary advantage allows you to survive and therefore reproduce. So we need to be able to say that they are fast, because that's why they're important. They're very rapid or they're very fast, and that allows protection from harm or damage. So it protects the body from harm or damage when it happens, and that means that they're more likely to survive, less likely to suffer injury or death, and so you're more likely to survive and reproduce, and therefore that's why it's evolutionary advantage to be able to survive these things. Okay, give 2 other examples of autonomic responses by the autonomic nervous system. So apart from pulling your hand away from pain, so not a pain response, give 2 other reflex responses. So these are things that happen automatically without you thinking about them, that can protect you from danger. They're not things like jumping out of the way of a car or being able to catch a ball if it's thrown at your face. Those you have to think about. Your brain has to process information in order for you to react in those situations. We're talking about kind of reflex reactions that are natural, you're born with them, and they don't involve the conscious brain. So things like blinking if something comes into your eye, a knee-jerk reaction when someone hits your leg and your knee kind of bends, pupil reactions, so contracting or dilating of your pupils in response to light, sneezing if something like pepper or dust or something goes up your nose. All of those are something that happen without you thinking about them, that happen quite quickly. So I'm going to go for when your pupils contract in bright light, because that protects you from harm, and when your knee jerks when your leg gets hit. 2 students wanted to compare the function of their nervous system by testing their reaction time using a ruler drop test. Hopefully we've recognised this already as the required practical for paper 2. There aren't many required practicals for paper 2, so it's definitely worth learning the few that there are. So this is the one where we're like using ruler drops to test our reaction time. So they used the following method. Student A held the ruler just above student B's hand. Doesn't say which hand, so that's kind of important, and we'll talk about that as one of the variables in a minute. Student A let go of the ruler without warning, and student B had to catch it with their thumb as soon as possible. The reading on the ruler where the student's thumb was in centimetres was recorded to show how far the ruler fell before it was caught. They repeated this 5 more times, then swapped over to student A was catching. So they are measuring the number of centimetres that the ruler fell through their hand before they caught it. And they did repeat the experiment, which is good. It's important to repeat experiments, we should know, and it means that we've got some sort of reliable results. Okay, so the actual question E says, state 1 factor the students could have controlled to make this investigation fair. Well, it's basically just asking what should be kept the same for both students. And so anything that should be controlled is something that can affect what you're measuring. So in this case, we need to think about all the things that can affect reaction time, and they're all the things that should be kept the same unless you are changing them. So they're not testing to see if there's any specific effect of tiredness or caffeine or anything in this practical. They're just looking to see what the difference is between the 2 students. So we need to think of things that can affect reaction time. So how tired you are, your age, your sex, how much practice you've had, how much caffeine you've had, whether you've used the same hand. So in this case, that's kind of linked to practice, but if you use a dominant hand, that's going to be stronger than a non-dominant hand, and you're used to catching things, so you'd expect your reaction time to maybe be faster with your dominant hand. So all of those can affect reaction time. I'm going to say the students should both use their non-dominant hand, but you could say they both need to be the same age, they both need to be the same sex, they both have to have not practiced before, so they're using their non-dominant hand, or they both had to make sure they hadn't had the same different amounts of caffeine that day. One had a coffee and one didn't, and that wouldn't be fair. So any of those that can affect reaction time we can use as a control factor.
Okay, so now we can see the results. After converting their ruler measurements to reaction times using a table, hopefully you remember doing this, so obviously the actual distance isn't your result, you need to convert that to the reaction time in seconds. So they've got the following results. They want us to calculate the mean reaction time for student A. Now this is a 2-mark answer. So first of all, how do we calculate the mean? Well, we add up all the numbers and divide by the number of values. However, if you're calculating mean, you should know that you should do that, but only do that once you've removed any anomalies. So we need to take out any anomalous results and then add up all the numbers and divide them by the number of values. So I'm looking at the list for student A. So we've got 0.27, 0.4, 0.23, 0.24, and 0.22. Now the 0.4 is jumping out at me immediately as a potential anomaly because it's nearly double all of the other values. And if you look over to student B as well, their highest was 0.31. So it's way higher than any number on the table, whether it's for student A or student B. So I would say that this is most likely an anomaly, given how close in range all of the other numbers are. They're all in the 20s. I would say we remove that, and so then we calculate 0.27 plus 0.23 plus 0.24 plus 0.22. That gives us a total of 0.96. And then we're dividing it by 4 because we only had 4 values, so that gives us 0.24. Okay, part H. Give one conclusion you can make from these results. Well, if you're looking for conclusions, especially in the table, then you need to look for patterns or differences that you can talk about. So there's a pattern that I can see, which is that for both students, the reaction time decreases as they have more goes. So it decreases with practice, which might be something that you looked at when you did your reaction time. And also we have a difference. So student A's mean reaction time is lower than student B, so they have a faster reaction time than student B. So there's a difference and there's a pattern. I'm going to go for the difference because that's kind of what they were investigating, was they were comparing their reaction time. Okay, so question 5. You need for mark 3 for 5A, you need to have the neurons in the right order. So they'll only be accepted in the right order. They say travels via, or the impulse travels via. That just means through or along. Either of those is fine. Should be the idea that it's going through these neurons or down these neurons. You need an electrical impulse. Can't really say signal. It has to be impulse really for mark point 2. But you can say the stimulus or the pain for mark 1, or the muscle or the effector. You don't have to say both. You can just say one or the other. That's fine. For 5B, you need to talk about the fact that it's increasing their chance of survival. That's what makes it an evolutionary advantage, because they're more likely to survive and reproduce and therefore pass on their genes. You've got lots of options for 5C. We talked about most of them. It's mostly thinking about the fact that these reflex reactions are true reflexes that you don't think about. It's not the same as jumping when you see a car coming or catching a ball when it's thrown at your face. Those are not reflexes because you use your brain for those. And then you've got, again, a lot of options for control variables. You can't just use the same ruler. In control variable questions, never talk about equipment, because really very, very, very rarely would something to do with equipment need to be controlled. In this case, you can mention the equipment, but it has to be specifically that the weight of the ruler is the same, because obviously that will affect the speed at which it falls. So you need to make sure that you don't talk about equipment if you don't need to. You can think about things that will affect the experiment. That's what you need to keep the same or control. Okay, so the last two parts of question 5. 0.24 was the mean, we explained why, but if you calculated the mean correctly but without removing the anomaly, but you still did it in the correct method, or if you did it in the correct method but somehow got the wrong answer, then you can have 1 mark out of 2. So really there's 1 mark for calculating a mean and 1 mark for removing the anomaly. And then for 5F, you've got these 3 options. So student had a faster reactions than student B. student A caught the ruler faster than the student B, or reaction time decreased with practice, which was the pattern that we noticed in the results. All of these aren't just saying like, oh, student A's time was faster. You just need to be careful about how you word it. So the conclusion is, because they were testing reaction time, we should have reactions or reaction time as part of our answer. Okay, the graph below shows how the hormone levels change during a normal human menstrual cycle if there's no pregnancy. So we've got FSH and LH and estrogen and progesterone, and the hormone levels and the days on the bottom. So the first question being, using the graph above, which hormone stimulates ovulation? Okay, so what does ovulation mean? Well, it's releasing an egg. So on what day do we release an egg? Hopefully we know that's around day 14. And at day 14, what kind of peaks of hormones do we have? We've got kind of estrogen is dropping, and then we have FSH and LH both have a peak, but the peak for LH is way higher. So it is LH that causes the egg to be released. Hopefully this is something you have learned and know, but that's kind of how I would work it out from the graph as well, is thinking, okay, well, that has the biggest peak, so that's causing what's happening on that day 14. What two hormones are produced by the pituitary gland? Okay, well, estrogen, we should know, is released by the ovaries, and so is progesterone. They are both released by the ovaries as their gland that secretes them into the blood. So the other two are released by the pituitary gland, LH and FSH.
Okay, part C. Explain how a sperm cell is adapted for its purpose of fertilising the egg. Okay, so we need the feature and the reason why it is able to swim to the egg, or how that helps it do its function. But you need to think about, it's 2 marks here, so you say 2 things. So 2 features linked to how it helps the sperm to do its job.
Okay, so I've got a diagram of the sperm cell to remind us what we're dealing with. So they have a tail to swim to the egg. That's a unique feature of sperm cells because not all eukaryotic cells have a tail. They also have lots of mitochondria. Now, the lots of is really important because every eukaryotic cell has mitochondria. So we can't just say they have mitochondria to release energy for movement, to move the tail, because that's what they need, they do. But if you said that, that's not an adaptation because all eukaryotic cells have mitochondria, but we're saying what is special about them is that they have lots of, or many, so lots of mitochondria release energy. It's important that we say release. We're not making, we're creating energy, we're releasing, we're transferring energy for movement. And then how do we move? Well, we have the tail, which allows the cells to swim. Okay, next part. Genetic sex of a human offspring is determined by the sex chromosomes carried in sperm. Draw a genetic diagram to show how sex is inherited. Okay, so in this context, when we're talking about sex inheritance, what we're talking about is obviously genetic sex being male or female. And it says that the sex chromosomes, so the X and Y, in the sperm determine whether you get male or a female. So if the sperm is the gamete that's determining the sex of the offspring, then it must have one of each chromosome. I hope that makes sense. So we should hopefully know that we have XX or we have XY, and that is male or female. So 2 X's is female, XY is male. The deciding factor is whether you inherit a Y chromosome or an X chromosome from the sperm. So that means that we should hopefully know that the sperm has both, or one of each, and the egg is XX. So what's a genetic diagram? Another way of saying that is a Punnett square. So what's a Punnett square? To show how we inherit sex. So a Punnett square showing the sex chromosomes in the sperm and the egg, and then what the potential offspring will be. So I'm just going to quickly draw my Punnett square out, sketch it out. Okay, so now we need to put the sperm chromosomes on one side and the egg chromosomes on the other side. So we have XY in the sperm, which we said, and then we have XX in the egg, which we said. And now we just have to fill in the offspring boxes. So you just make sure you've got what's in the left hand and the top box for each square. And then we should know, or hopefully know, that this is correct because it means that we have a 50-50 choice. So we have 50% of the boxes would be XX and 50 would be XY. So that means it's correct because sex inheritance is a 50-50 chance. And yeah, you need to make sure you've written the sperm and the egg really clear and next to the correct chromosomes around this Punnett square. To prevent pregnancy, contraception can be used. The table below shows some information on different forms of contraception. So we have the pill, we have condoms, we have the IUD, and we have sterilisation. And then we have the effectiveness, which is 99% for the pill, 98% for the condom, more than 99% for the IUD, and 100% for sterilisation. And the question is, evaluate the use of these contraceptive methods for 6 marks. So it's an evaluate question, which means that we need to do the pros and cons of each, so advantages and disadvantages of each, and we're going to use the effectiveness and the other information to help us find that out. And then we need to end with a judgment. So that means your opinion on which one of these is best, justified with some points as to why you think it's best. It doesn't really matter what your opinion is, as long as you've backed it up using information that you've been given in the question. So I think this is the best method of contraception because, and then give a couple of reasons why, based on information you've got from the question. Okay, so we've got a lot to talk about here, and there's loads of things we can say. I'm going to show you how I'm going to kind of put them in comparative little groups. So I'm talking about the advantages, the disadvantages of the pill, but as I'm doing that, I can then think about, can I link some things together here? Because it's going to be linking that's going to help us to get high marks in this leveled answer. I'll go through the leveled mark scheme with you as well at the end. Okay, condoms are the least effective here, but are still 98% effective if used correctly, and they're the only method which can protect against STIs. So I've given some positives there. However, some people may not be able to use it if they're allergic to latex. Negative. Both the pill and IUD are very effective at over 99%. Positives. However, this is only if people remember to take the pill at the same time every day, and it can cause side effects such as headaches. Negatives for the pill. The IUD is a long-term contraceptive. Positive. But it can also cause pain and there's a risk of infection. Negatives. Surgical sterilisation is the most effective method at 100%. Positive. However, it's permanent, so this is not an option for people who want to have children in the future. Negative. Overall, I think that condoms are the best contraceptive solution because they protect from STIs, they are not permanent, and they do not have side effects, but they are still very effective. There we go. And I've given a judgment on condoms. So I've given the best things that I have about them, comparing them to the others. So they're the only one that can protect against STIs. They're not permanent, like sterilisation. They don't have side effects, like the pill. And they're very effective at preventing pregnancy because they're still 98%, which is still a lot. Okay, so there's my 6-marker paragraph, and that's how I've written it. We'll go through the mark scheme as well so we can see what we actually get marks for. Okay, so part F. If a person is having trouble getting pregnant, they may start infertility treatment. Describe the process of IVF. So IVF, in vitro fertilisation. Describing the process, as we've already said, describing a process means doing the steps in the right order and making sure we talk through the detail of what happens in each step. We're not being asked to explain why here, so we're just describing what happens. So I would quickly just jot down the main steps of IVF so we make sure we cover them all and we write it out in full. So the woman needs to produce eggs. In order to do that, they have to inject hormones, which is LH and FSH. The eggs will be harvested. They will be fertilized outside the body, so in vitro, using sperm. So they added to sperm in order to fertilize the eggs. And then they return the embryo to the uterus. So they're the steps in order. We've got to mention hormones. We've got to mention combining with sperm and fertilization. And then we've got to mention returning that embryo to the uterus to develop. Okay, so the woman will have hormone treatment with FSH and LH. This will stimulate egg maturation and release. The mature eggs are collected and fertilized with sperm. an embryo or fertilized embryo is then placed back into the uterus to develop. So we've said all the things. We talked about the hormone treatment. We talked that that's going to produce eggs or stimulate egg maturation, so it's going to mature and release the eggs from the ovaries. We're going to collect the eggs, we're going to fertilize them with sperm, and then the fertilized embryo is going to be returned to the uterus to develop. Okay, so mark scheme for this question, as we said, lots of needs to be in there to get the mark. So, and also we have to get the release or transfer of energy correct. So you can't have the mark if you didn't say release or transfer the energy. You also can't have the mark if you didn't say lots of or many mitochondria. You can't talk about energy being created because energy cannot be created or destroyed. So just be really careful with your language around that. So you get 1 mark for the correct gamete chromosomes. That's the ones from the eggs and sperm. So have to be labelled with sperm, have to have X and Y. Have to be labelled with eggs, having XX. That gets you 1 mark. And then 1 mark for the offspring. So that gets you the 1 mark for the correct X's and Y's in the boxes, the 4 boxes in the middle. As long as they are correct, then you can get mark number 2. Okay, so for the last question, there's a lot here. We've got a levelled answer. So you get nothing for no relevant content. You will get 1 to 2 marks if you made relevant points, but they weren't linked. You'd make some logical points and a simple judgment, but without justification gets you 3 to 4. And then a judgment strongly linked and logically supported by a sufficient range of correct reasons gets you to 5 to 6. So if you look down this bottom section, we mentioned a lot of these to show the kind of linking and the comparisons when we talked about our judgment at the end, or we mentioned them as we were talking about our advantages and disadvantages. So just giving a few advantages and disadvantages for some of these would get you 1 to 2 marks. Giving some of these as advantages and disadvantages for these, plus a simple judgment that just said, oh, I think condoms are the best, would get you 3 to 4 marks. And then an answer like we did, where we strongly linked our judgment to a range of reasons that were correct, because we talked about them and they were in the table, that gets us into the 5 and 6 bracket. Okay, so there's 4 marks available here, but there's technically 5 things you can say. So the woman is given FSH or LH, and you can have either of those. You don't have to say both like I did. To stimulate egg maturation, release, or production, you can say any of those. You don't have to say all of them. Eggs and sperm are collected or mixed, so eggs and sperm put together. Fertilization happens, so added to sperm or mixed with sperm, however you want to say it. Making sure you say fertilization, that gets you a mark. And then the embryo or the embryos are inserted into the uterus or womb. You can have either word there as well. So there's kind of a couple of options there that you've got that you can get marks for in this. But it's sort of very important as well that it goes in the right order and you explain steps. Okay, so that's the end of this paper. Hopefully that was helpful. And hopefully now you've kind of gone through the content for paper 2, seen some parts of this, whether you're more confident or less confident. If you've been less confident with this or you've missed keywords or marks when we've answered some of these questions, make sure you add those to flashcards and go back over the content so you make sure you have it there, especially if it was to do with keywords. And hopefully this has helped you target your revision for the real exams. Good luck.
Ouch! This is why in some videos I have unexplained scratches.